Hello everyone and welcome to Pocket Size Bio, Dr. Casco. Today's video we're going to continue chapter 6 with section 6.5, mitochondria and chloroplasts change energy from one form to another. So let's get to it. Looking at the first subsection here, energy transforming organelles, mitochondria, chloroplasts, and peroxisomes. Very, very critical organelles for the cell's functioning for energy transformations. So when we talk about energy conversions, we normally think about mitochondria, one of the main organelles in cells, and chloroplasts, particularly to photosynthetic cells and photosynthetic organisms. You have chloroplasts that also do some form of energy transformation. We know this already from elementary school, high school, and so forth. Okay, we're going to take a deep dive into these, both of these organelles in a lot more detail. Okay, so mitochondria, it's basically found in every eukaryotic cell, right? They are the sites of cellular respiration. Now, this is a complex process. We normally think about cell respiration as carbon dioxide and water yields, sugar, my mistake there. Sugar and oxygen yields carbon dioxide and water, and energy, ATP production, right? But it's much, much more than that. It's a complex biochemical pathway where you have a series of chemical reactions that are occurring, right? Ultimately to generate ATP from organic molecules. You also get chloroplast. Chloroplast, again, this is another energy transformation organelle. It's found in plants and algae, and these organelles facilitate photosynthesis, right? Where you have the conversion of solar energy into chemical energy stored in organic molecules, stored in glucose. Okay, so before we get into that, we're going to talk about how a theory on how how eukaryotic cells came about. Okay, mitochondria and chloroplasts evolved from prokaryotic cells that were engulfed, right, by an ancestral eukaryotic cell. This is called the endosymbiont theory and evolution, focusing on the concept of evolution, where we see in this picture here, you have an ancestral eukaryotic cell. So you have this primitive form of eukaryotic cell that was relatively large, right? Had some foldings inside the body. It had a kind of nucleus already forming, and you had this folding of the nuclear envelope. You have DNA that was housed inside this structure, but you don't see any energy, processing organelles or structures yet. So it is thought that, engulfing an oxygen-using non-photosynthetic prokaryote, but it didn't digest it. It engulfed it, but it didn't digest it. These smaller prokaryotic cells stayed inside this larger heterotrophic cell, right? And these little prokaryotic cells were quite specialized in processing oxygen. Over years and years and years, they developed a symbiotic relationship where these prokaryotic cells ended up, providing necessary energy and materials to this other larger cell. Okay. When you get a photosynthetic eukaryotic cell, a similar event happened where you're getting an engulfment of a photosynthetic prokaryote that is able to process light energy into chemical energy. It was engulfed but not digested, and it stayed in there, and you get this rise of a photosynthetic eukaryote. Now, there's some evidence that does support this idea, where both organelles Basically, the chloroplasts and the mitochondria are double membrane. We're going to take a look at that in a bit. Okay, they're double membraned organelles. So they have two layers of the phospholipid bilayer surrounding them. So that's an idea that will suggest their prokaryotic origin. You also have that these organelles have circular DNA and their own ribosomes within their body, within their structure. And that is a kind of signature feature of prokaryotic cells. And they're also semi-autonomous. So what does that mean? Essentially, they can divide, they can reproduce and grow independently of the larger this larger prokaryotic cell, which ends up being the eukaryotic modern cell that we think about, right? So if you have this this host cell, right, it divides independently of the rest of the cell. Okay, so these are lines of evidence that can support this endosymbiotic theory over time, right? To give rise to a heterotrophic eukaryotic cell or a photosynthetic eukaryotic cell. All right, so let's continue more to the specifics of mitochondria. I do apologize for my throat. It's a little hoarse today, but nevertheless, it's over. So mitochondria, chemical energy conversion, right? So it's found in nearly all eukaryotic cells, okay, which range between 1 micrometer to 10 micrometers and can vary in the amount of the quantity that they're doing and the amount of processing that they're doing at a given time. There's a relationship between the metabolic activity that that the cell needs and the activity, of course, of the mitochondria. Okay. For example, cells that move and contract would require more mitochondria, of course, for energy processing, for ATP production. It is critical that, I believe, for undergrads and even high school and AP biology, you must master the whole structure of mitochondria. Okay, so you have the structure and its components. I'm going to zoom in a little bit. You have an outer membrane that is relatively smooth and serves for protection. Okay, you have this outermost membrane. You have an inner membrane. Okay, and that inner membrane that you can see here essentially folds, increasing the surface area, right? So that inner membrane and those folds are referred to as cristae, okay? And it functions to increase the surface area for protein production, for ATP production. In between these two membranes, right, you have a intermembrane space, and it does involve some movement of hydrogen plus ions in this whole process of ATP production during the electron transport chain. Okay, and within the inner membrane, that space within that inner membrane, you have the mitochondrial matrix. Here you just have a big soup of mitochondrial DNA and enzymatic proteins and ribosomes. Okay, these are the main structures. Make sure you're familiar with the structures. Okay, the back of your hand. They generally have a little bean shape, but they don't necessarily have to. What's important is that you can identify under the electron microscope these cristae. If you see all these cristae that are forming, okay, to enhance metabolic activity, right? Chemical reactions to occur with a lot more efficiency. Okay. Again, function. Diving a little bit deeper into what it does, it undergoes a series of reactions, essentially cell respiration, right? Using oxygen to break down glucose and achieve ATP production. Okay. Again, as I mentioned before, the cristae enhance ATP production during a particular moment called the electron transport chain. If you have more surface where you have all these proteins embedded on the membrane, then you can be producing more in a particular time. Okay, that we're going to see during the energetics chapter. So don't worry about it for now. Just understand the essential function that it does. Okay, mitochondria are very mobile. They can move around. They can change shape. They can bud and divide, and even make a fusing network, right? For the whole purpose of producing more energy, depending on the necessity of the cell. Okay. So mitochondria, very important to know the structure. Right. Let's move on to chloroplast. Chloroplast, again, another organelle that is double membrane, and it functions for capturing light energy. Okay, you can see its general shape. Yes, it is a lens-shaped organelle. It's around 3 to 6 micrometers in size. And particularly, why is it green? It has a green pigment, chlorophyll, and this chlorophyll molecule is very important for that absorption of light, right? Along with other enzymes, it works to produce sugar, right? Works to produce sugar. Again, some compartments and structures that you need to know. With the basics, the outer and inner membranes, same idea here. It encloses the outer membrane, encloses the entire organelle. Okay, the inner membrane also, you have that intermembrane space between the two membranes. The inner membrane houses what are called thylakoids and the fluid within that space. These thylakoids are very, very important. They're little flattened sacs of membrane themselves. If you take a look here, they look like little discs. Okay? Little have balloons that have been kind of squashed into little pancakes. Okay, so each of these little discs are called thylakoids, and they could stack up together, and they can interconnect together from one stack to another. And these stacks are referred to as grana. I'm going to zoom out a little bit. Okay. And a particular type of reaction occurs here that is dependent on light. It's referred to as a light-dependent reactions. They're occurring on the membrane of these thylakoids, of these grana. Okay. A similar process occurs here to mitochondria, which is the ETC, right? The electron transport chain, but we'll see that in the later chapters. The fluid in which the thylakoids are kind of embedded in is referred to as the stroma. It is a fluid surrounding the thylakoids. And again, here you have the chloroplast DNA, you have ribosomes for protein production, and you have your enzymes necessary to do these chemical reactions. Okay. So you have 3 compartment spaces. You have that intermembrane space, you have the thylakoids, and you have the stroma. Okay, inside the thylakoid, there's also a space there. It's referred to as lumen. Okay, so 3 spaces, compartment spaces. Chloroplasts are very flexible, right? They can kind of stretch out. It's very changeable in that sense, and they can move along even with the cytoskeleton. You probably have seen this from high school and middle school, cytoplasmic streaming, that these little chloroplasts can move throughout to help enhance light capture, right? Just want to mention about the electron micrographs here, as it could be questions in tests, right? So you have your chloroplast. These dense regions are the grana. Okay, this is quite a signature coloration here. And amongst these little thick structures, you have fibers that are interconnected amongst them, okay, for communication and passing of material. All right. Here's an example of an algae. If Spirogyra specifically, this is a type of algae where you can see chloroplasts running along throughout the structure of an algal cell. to enhance photosynthesis. Okay. Again, main purpose of chloroplast is to convert solar energy to chemical energy via photosynthesis. So it's producing sugars and other organic molecules for plant metabolic activities. Now, it's quite interesting that you don't see this often in high school biology, but there's more than just chloroplasts in terms of organelles. There's different types of organelles that belong to this group that the chloroplast belongs to. They're referred to as plastids. And you have another type of plast, right? A meloplast. You have this structure that can primarily store starch, okay? A kind of stored form of glucose, okay? Referred to as a meloplast. They're more globular in structure. And you have chromoplasts. Chromo, you can get an idea of what it's referring to as the coloration, right? Here, you have these structures that contain pigments that are found in fruits, that are found in flowers, that are not necessarily green, right? They have this brownish, reddish, orange pigmentation that provide the color of these different structures in the plants. Okay? Chromoplasts. So for now, that's all you need to know of chloroplast structure and the general specific structures and the general function. Peroxisomes, again, another structure that is involved in energy conversion or breakdown of material. Okay, one of its essential functions is for oxidation and detoxification. Right, it removes hydrogen atoms from substrates, transfer them to oxygen producing hydrogen peroxide. Okay. Now, this is a single membrane structure. And you can see this here. This is a peroxisome. It does have several functions. Okay. But essentially, it uses oxygen to break down fatty acids. These fatty acids could then be used by the mitochondria to for energy, okay, as a molecular fuel. The fatty acids are a good source of energy. It could also detoxify harmful substances. So alcohols, the OH groups in the alcohol, right? They can be broken down. They could be converted to produce hydrogen peroxide, and further breaking it down into water. Okay. This is more particular for the liver, right? It detoxifies harmful substances. Okay. Ultimately, it's the hydrogen peroxide we want to get rid of, okay, to break it down into water. There are other types of peroxisomes. So you have glycoxysomes, okay, glyocysomes. Sorry. They're found in fat-storing tissues of plant seeds, okay, and they convert fatty acids into sugars to support the early growth of the seedlings. Okay, of course, seedlings don't have any pigmentation to undergo the light reaction, so they need another form of energy before up until photosynthesis. The photosynthesis starts activating. Okay. Interestingly, though, the evolutionary origins and replication mechanisms of peroxisomes remain unclear. So there still is a good area of investigation, right, on how these structures came about. Okay, peroxisomes. Let's continue to 6.6. All right, this is the cytoskeleton is a network of fibers. So we move from energy to structure. It is a network of fibers that organizes the structures and activities of the cell. The key component here is the cytoskeleton. So we're going to look at the structure and the function and components of the cytoskeleton. Here's an example of what the cytoskeleton looks like. This is a micrograph using fluorescence. Okay, and what you're seeing here is that several components of the cytoskeleton is being tagged with fluorescent molecules. And you can see here, green is for microtubules. This is one type of subtype of cytoskeleton. And reddish orange for microfilaments. Very, very effective for biologists that are studying the structure of the cell or how a cell is changing in its amount of cytoskeleton at particular times in its life. Okay. So let's take a look. All right. Early microscopy, right? Biologists thought that eukaryotic organelles floated freely in the cytosol, like a big pool, right? They're just floating randomly. But there's good evidence now, and of course we see it with clarity about the cytoskeleton that is ultimately a network of fibers that can hold and they can extend and retract and keep the certain structure of the cell, even move organelles from one place to another, move vesicles from one place to another. So it's kind of like a highway system. Okay. Ultimately, you have this network of fibers that is extending through the cytoplasm. Okay. Bacterial cells and prokaryotic cells do have a cytoskeleton, right? Cytoskeletal fibers, but the book emphasizes more on eukaryotic cytoskeleton. Okay. In terms of function of support and motility, right, there is an emphasis on mechanical support to maintain a cell shape, especially with animal cells. We don't have cell walls like chloroplasts, plant cells. So how do we maintain that rigidity for each individual cell, right? So we have the cytoskeleton. Okay. This is a nice picture also that kind of emphasizes the idea that of support. The cytoskeleton produces like a dome tent, and you can see that here. Okay, and ultimately that helps balance forces, right? So you should have different forms of pressure, the external pressures and internal pressures of the cytoplasm, and you need to have something that can withstand and balance out these forces. Okay, as I was mentioning before, the cytoskeleton can anchor the organelles, cytosolic enzymes, molecules, large proteins that need to be in a particular place or move within a particular area. Okay, they can help transport them around. Okay, so it's highly dynamic. The fibers themselves, depending on how the cell needs to move about, particular cells, it can disassemble, reassemble. The edges of these fibers can break down to a certain extent or regrow, reform also. Okay? Disassemble and reassemble. Cytoskeleton also is useful for cell motility, like I mentioned before, where the entire cell can just move about because of that extension of a fiber going in one direction. Maybe there's some signaling stimulant that is enabling the cell or making the cell move toward that particular direction. Okay, so it helps in movement. It's very, very interesting for this next part. The fibers, we're going to take a look at the fibers more specifically, can work with motor proteins. So these motor proteins, they literally look like little feet. And these proteins, as you're using ATP, one little part of the protein can move forward, and the other leg of that protein can move and move and move. And as these proteins are moving along, they can actually carry vesicles, structures along with it. So they're pulling and moving and walking a particular thing from A to B. Okay, very, very interesting. So motor proteins, they facilitate intracellular transport. Vesicles, an example as you see in the visual here, vesicles use motor proteins to walk along cytoskeletal tracks, right? especially, using vesicles of neurotransmitters, um, uh, where you're going to use them in nerve cell, axon terminals. Okay. Not just that, you also assist in plasma membrane modifications, forming food vacuoles or phagocytic vesicles. Okay, so it's always being used consistently, constantly throughout the cell's life, even during cell division, especially during cell division, as we're going to see later. All right. So the components of the cytoskeleton, there's 3 types. And the book has a very nice summary. I'm just going to zoom in really quick so you can see the table in its entirety, but I've clipped it so we can focus on one particular one at a time. Table 6.1, make sure you study this in its entirety. This is very good. Structure and function of the cytoskeleton. So there's different types: microtubules, microfilaments, and intermediate filaments. So this is the biggest, this is the smallest, and this is the kind of middle ground type of fiber. You can see their how big they are, right? In terms of diameter, 25 nanometers with 15 nanometers of the lumen. Okay, so it looks like this to give you a reference. 7 nanometers microfilaments, okay, and 8 to 12 nanometers, this intermediate filament that you see here. So we're going to go one by one. Okay, but you do need to memorize these 3 types. Okay, microtubules, I'm just going to zoom out a little bit here. They're present in all eukaryotic cells. Critical protein, you must memorize this. They're composed of tubulin. You're going to hear this word a lot, especially in research. Tubulin, alpha tubulin, and beta tubulin are proteins that create this fiber. If you see here, you see the lighter coloration of this protein is alpha tubulin, beta tubulin, and they work together. They produce a dimer, what is called a dimer, an interaction between these 2 units. And in research, they're used a lot as a control, right? Like EF1 alpha. They're used as control markers, right? For PCR, if there's a change in expression of a certain gene, these proteins stay the same pretty consistently. So they're often used as reference genes because every cell pretty much has them. Okay, so they're built from tubulin dimers. So you have a alpha tubulin and beta tubulin protein that are working together. Very interesting. They're highly dynamic. So the edges, if you see here, can disassemble. So those proteins can disassemble, shortening the microtubule, or it can extend. They can grow and shrink by adding more dimers of these proteins. Okay. Yeah, there is a particular end. You have this plus end where assembly and disassembly occurs at higher rates, okay, enabling that extension and so forth. Ultimately, the function for this type of fiber, of cytoskeletal fiber, is to provide shape and support. It also serves as a kind of superhighway track for proteins to move along and to transport organelles to kind of slide through the cell. And as we're going to see in the later on during mitosis and meiosis, for chromosome separation during cell division. So they're heavily working during this process. Particular structures that form from these microtubules are centrosomes and centrioles. Okay, in animal cells, you have microtubules. They grow from a centrosome. So you have this kind of region, okay, that is composed of 2 structures that are called centrioles. Okay. You can see them here. This is one centriole. This is another centriole. And these are structures that consist of microtubules that are assembled in a very unique way. Okay. And just want to see if I have that. arrangement. We're going to talk about that arrangement in a little bit, but let me just go back here. Okay, the centrosome, it has two of these centrioles, and these are structures that are made up of 9 sets of triplet microtubules. So as I zoom in, you can't see it that well, but you can see there's one fiber, there's another fiber, there's another fiber, and they're arranged in this particular order, okay, in 9 sets of triplets, okay, triplet molecules. So these centrioles, they help organize the microtubule assembly. Some eukaryotic cells do lack them, as they don't require them as much because they can be space consuming. Okay, if you see them under a microscope, right, a micrograph, electron micrograph, more specifically, this is the arrangement that scientists have been able to discover. They're arranged always in this kind of perpendicular form. One is going vertical and one is going horizontal. Okay? Sometimes they're not even present depending on the stage of the cell. Whenever they are needed during cell division, they are prominent. Okay. Let's continue. So these microtubules, many functions, right? So the first one for that cell division, for just cell support, they can also be found in cilia, in organelles and organisms that are moving around, cells that are moving around. You have these special types of structures. They're referred to as cilia and flagella. These are extensions of the cell that contain microtubules. They specialize in movement. Okay. You can see them in, of course, structures that need to move about. Unicellular protists, single-celled protists, animal sperm, some plant cells, spores. You can see them move about. Okay. And for flagella are structures that are a few per cell, right? Generally one long kind of tail that moves in an undulating motion. Okay, similar to a fish, you can see this direction of swimming motion of flagella. They're generally quite long. Okay, we see this is the body of this particular structure here. Okay, and you see how long that flagella is. They're fewer, but they're a lot longer, stronger. Then you have cilia. They're short and numerous. You can see many filaments here. Okay, and in this particular case, you have, let me just move over here. Okay, you have these little hair extensions that are on the surface of a structure of a cell, right? And they can be used for several things. You could use them for attachment. Okay, you can use them to sweep material along, let's say, your respiratory tract. We have a lot of cilia in the trachea to sweep mucus and bacteria and debris out of the lungs so you can expel. So instead of having this undulating motion, they tend to have a sweeping motion where you have a power stroke, you have that forceful movement, and then you have a recovery stroke, which is a lot contains a lot less force. Okay, you have this power stroke, and then you have a little recovery, softer stroke. Okay. There are some cilia that are not particularly used for movement, but they can receive signals. There's even some specialized cilia that is used for reproduction in bacteria, right? Conjugation. They've become specialized for transmitting genetic material. Okay. Yeah. Non-motile cilia, so they help in communication, right? Signal receiving antennas of the cells. They transmit environmental signals to initiate pathways, intercellular pathways, signaling pathways. Okay. Brain function and embryonic development, very important. Again, as I mentioned before, oops, I apologize for this. We don't need that in our notes. So the structure of the cilia goes in a 9+2 microtubule arrangement for cilia and flagella. Okay, if you see here, this is a little bit different from the centrioles, okay, but you have this ringed arrangement, okay? So there's 9 outer doublets with 2 central microtubules. So we see here, okay? Here are the doublets, and then you have 2 here at the center. All right. So this is cilia and flagella. At the base of these filaments, you have a structure similar to the central arrangement. These are called basal bodies. Okay. So if we take a look, let me zoom in here. Why am I not zooming in here? You have these particular structures. You zoom in into a micrograph. You can see the arrangement of the doublets. But then at the base, to have more rigidity, more stability, you have this basal body, which is the triplets that we know about. Okay. An example of these particular proteins that would use these is dynein. Okay, dynein, motor proteins. They use ATP to create the bending movements walking along the adjacent molecules. Okay, good. Let me see if I'm not missing anything here, just for you guys to have everything covered. Okay, let's continue on. Okay, moving on to the smaller extreme. To the small extreme, you have the microfilaments. These are filaments that are made out of actin proteins. Okay, they're solid, thin rods that are composed of this particular protein. Okay, and these filaments, let me just zoom in a little bit here. They chain up, right? They chain up and they can actually twist among themselves to create this microfilament. They form twisted double chains and they create structural networks when they're bounded by themselves and with other proteins. Okay, they're also found in all eukaryotic cells. They provide tensile strength and they resist pulling forces. Okay, and moreover, they help provide support for the plasma membrane. Okay, for the plasma membrane in particular. Okay. They're referred to whenever they're in support to the plasma membrane. They're called cortical microfilaments. So they help maintain also the cell shape, okay, and give the cytoplasm that gel-like consistency also. Not just water, but the cytoplasm is quite jelly-like, and it has that thicker consistency because of this microfilament. Okay. They also help in the extension of intestinal microvilli. These are extensions on our intestines, on our small intestines, to increase surface area. Okay, if you take a look here. Okay, you can see the little microfilaments here. Okay. And they help, of course, keep these little filaments upright for more absorption. More absorption means more nutrients for us whenever we eat. Okay. Just go a little bit down here. Movement, a role in motility. So here you have actin filaments. They interact with another protein. Okay, we're going to see this a little bit later on when we get into muscle. But just a general gist here. They interact with myosin proteins to facilitate movement, and they tend to kind of force a movement so the actin filament can contract the myosin filament. Okay, so there's a very unique interaction among these two proteins. And that's how muscle contraction actually works. Okay, contracting and relaxing. Contracting, so they just contract there. Okay. Here you can see a schematic of muscle cells, right? Or so you have actin filaments, you have these myosin filaments, and they have these little bud structures, and the bud structures, they create a movement down, and they actually pull the actin filaments closer, and that movement to each actin filament becoming closer will create the contraction that we know of as a muscle contraction. Okay. They also work again, not just in muscle contraction, but amoeba movement to create that pseudopodia, those extensions to move around. Okay. They also help for plant cells. So actin contributes to the cytoplasmic streaming that I was talking about a little bit earlier, for the movement of chloroplasts and just to distribute material more effectively. Okay. Here's an example of the visual of the amoeboid movement. Okay. Just by kind of manipulation of actin filaments, you can extend part of the cytoplasm and the cell membrane of these little organisms to go a particular way, and so forth. Okay. And this is an example of cytoplasmic streaming where you have the materials circulating. Okay. Going on to intermediate filaments. Intermediate filaments, as I mentioned before, the name tells you like their relative size. They're in between microtubules and microfilaments. They're found only in some animal cells, including vertebrates. Again, this is a structure that does help provide tensile strength, okay, more stability for the organism, for the cell. Okay, these different types of proteins can help create this type of filament, but predominantly it's the keratin. Okay, and they're a lot more durable, they're a lot more stable than microtubules and microfilaments, and even persisting after death. So you can see these structures still there. Okay, keratin filaments. The outer skin consists of dead cells reinforced by keratin filaments, right? So keratin, very, very important to have, very, very important to make. That's why it's so useful in kind of health cosmetics, right? To maintain a healthy cells, especially on our skin, right? Skin and hair and so forth. Okay. So more specifically, the functions: it reinforces cell shape, fixes organelle positions, helps the nucleus be in its location, keep its rigidity, keep its shape. Okay. As I mentioned before, when I was talking about the nucleus in the previous video, it forms the nuclear lamina, right? If you remember, the nuclear lamina is that kind of network of fibers. Now you know that they're intermediate filaments, that lines, that coats the whole inner side of the nuclear membrane, of the nuclear envelope, more specifically. Okay, so it helps provide a permanent framework for the entire cell. All right, almost done with this video. The next thing we're going to talk about is more particular to plants. Okay, not necessarily, but we're going to take a look. Extracellular components. So we get out from the fibers to what's outside of the cells. Okay, extracellular components and connections between cells help coordinate cellular activities. So cell walls. Let's begin with cell walls of the plants. We can see here, we all know from secondary school, cell walls of plants, they're there for structure support predominantly. They help protect, serve as a barrier against the external environment for plants, and they also help for water regulation. Okay? The cell walls are actually composed of two types of walls. You have a primary cell wall and you have a secondary cell wall. Okay, and you have a space in between them, which is called the middle lamella. Okay? And these cell walls are composed primarily of cellulose. And we know cellulose now from previous chapters. They're microfibers, right? They're fibrils embedded in a matrix of polysaccharides and proteins that make it very rigid, very stable. Okay. Primary cell wall, and take a look here, right? The primary cell wall is that orangey looking one. The secondary cell wall is that brownish appearance one. It's a lot thicker. Okay. Primary cell wall is thin and flexible, and it's secreted first. It is what's created first. Okay. You have that middle lamella between each of these little structures, right? It's rich in pectins. Pectin is a very special molecule that helps glue, is very sticky, helps glue adjacent cells together. Okay. And a secondary cell wall. This secondary cell wall deposits, it's made after the cell grows, and it provides additional strength. Okay, but overall, this orange and brown is the cell wall. Okay, but it's divided into two parts there. You can see a micrograph here, much better. All right, you have your orange, and then the second one grows inward. The secondary cell wall grows inward. The lamella touches between the two different cells. That helps kind of glue them together. Okay, this is for plant cells. For animal cells, we don't have this structure to help support, but we do have what is called an extracellular matrix. This is called an ECM, and it's contained of a lot of fibrous structures composed of glycoproteins. You have collagen, you have proteoglycans, you have fibronectin, okay, that we're going to take a look at later on. In particular, that I do want you to remember is collagen. Collagen is a glycoprotein that accounts for 40% of human protein content. It is so important to our structure, to the integrity of the cell and the external environment. Okay. Fibronectin binds to other proteins that are referred to as integrants. These are transmembrane proteins. Remember what transmembrane means, that link the ECM to the cytoskeleton. So if you take a look here, okay, you have your extracellular matrix here with extracellular fluid. Okay. And these transmembrane proteins are the integrants. Okay, they're membrane proteins that help interconnect one with another there. Okay. Again, you have surface proteins, fibronectin, that helps attach the ECM to integrants. So they're all working together. Apologize with the bell there. All right. Very, very important. So the ECM does help regulate materials coming in and out. It influences gene expression, cell behavior, and even tissue coordination. Okay. Lastly, for this chapter, what are called cell junctions. So these are sites in which one cell is interacting with another cell. Okay. You have plasmodesmata. Plasmodesmata are sites or channels, right, that allow cytoplasmic exchange between the cells, one plant cell and another. Okay. Sorry about that. I just wanted to see if I had another visual for plants. Plasmodesmata. It helps pass cytoplasmic material. You have tight junctions. This is more for animals. These are very, very important to help cells maintain each other in a tissue form. Okay. Cells neighboring it's a seal neighboring cells to prevent leakage. They're referred to as tight junctions. You also have desmosomes. Sorry about that. Okay, and these act like anchors, desmosomes. And you have gap junctions. So this allows communication between cells. Let me just see if I'm not missing anything else. Okay. So you can get a nice visual here. Okay, you have tight junctions. kind of knitted together, one cell to another. Desmosomes, where you have very, very, very tight anchoring. You definitely won't be able to let go of each other with this level of anchorage. And then you have gap junctions. Here, this enables cytoplasmic channels. You have movement of molecules and so forth. Okay, so they help communication and they help keep tissues together. Okay, having tight junctions prevents leakage and movement of materials to get in between. Instead they'll go where they need to go. Okay. So ultimately, cytoskeleton and the ECM work together to maintain a general stable structure, facilitate movement, material coming in and out of the cell, and coordinate cellular activities with signaling proteins, let's say on the cell membrane. Okay, we're going to talk about that in the next chapter. All right, just to kind of summarize now, a cell is greater than the sum of its parts. This is a very nice, very interesting way to see emergent properties, right? You can't have organelles together just randomly floating in a solution. They work together, they communicate with each other to create a more sophisticated structure, which we know is the cell. Okay? Cellular components work intrinsically, intricately, right, to carry out a particular process. Okay? There's a nice example of a macrophage. Macrophages engulf bacteria using actin filaments, right? They extend their cytoplasm and engulf anything they need to, like bacteria. They digest them with lysosomes. We know what lysosomes are now, to break down that material, these bacteria. They need, in order to break down that material, they need ATP from mitochondria, another organelle inside their bodies. Okay, how it actually works is from the genetic instructions coming from DNA, which enables a sequence of protein relay signaling pathways for it to ultimately carry out its function. So the cell's internal organization and architecture enable it to function as a coordinated, synchronized unit greater than the sum of its individual parts. Okay? Now, lastly, I want to say, you will see this a lot in the textbook, how they visually represent the molecular machinery in the cell. Become familiar with this type of diagrams. You have a cell membrane here. These are 3D rendered models that quite accurately show how it would look like if you were to be there and see it. Aquaporins, right? One type of protein that is on the membrane, calcium channels, proton pumps, receptors. We're going to take a look at all these in the next chapter. If you were to see how an actual cell looks like with all of these things we've talked about, it would look something like this. Okay? You have a chloroplast. Inside the chloroplast, you have the thylakoids and the granum, and you have all the proteins embedded within this to make the light reactions. You have the mitochondria, and you have the electron transport chain on the crista, right? So there's a lot of things going on. But nevertheless, in seconds, you can have a signaling pathway effectively go from one place to another in a matter of seconds, right? Even though there's just so much clutter that you think you see, right? But it's actually quite organized. These are examples of cell respiration in the electron transport chain that we're going to take a look at. There's an example of photosynthesis and the light-dependent reactions that we're also going to take a look at. They're quite similar, okay? Just to get a visual, right, of what is happening here. Don't worry about learning this right now. We'll go in the later chapters when we get into energetics. Okay. So here I kind of compiled a double picture here because this is a two-part of the book, 2 pages of the book. So this is a whole section of the DNA here. Just to visually see, this is the nucleus. At a nucleus and the nuclear pore, you have these incredible structures here that regulate material coming in and out. Okay. Here's the motor proteins I was talking about as they walk along these fibers, the microtubules. They're carrying a vesicle. That vesicle needs to go somewhere. They move it along in that particular area. Okay. So it's a quite interesting, quite beautiful form of the cell membrane, right? And this is just a cross section. Remember, this is a 3D structure that is happening. All right. So that's it for chapter 6. Okay, I'll give you a look at the chapter review, guys. It's always good to read the concepts, the summary of the concepts. You can pause it to give you so you can extract the notes there if you need to. You move on to chapter 2, so, page number 2. Okay. All right. Thank you, guys. In the next chapter, I'll be talking about chapter 7, where we get into the details of membrane structure and function. Incredible things happening at the cell membrane. Here you can see some exocytosis or endocytosis occurring at this 3D model, at this kind of computer model of the cell membrane. All right. Thank you guys for watching. I'll be back with another one.